So, morning we have discussed about the structure of the heme, and as I already said that one point I have missed in the last before class, that is in heme the iron atom is present zero point 0.4 angstroms outside the plane. Upon oxygenation in the fifth coordination, in sixth coordination site, the iron atom moves into the plane. That is the first conformational change that is observed during oxygenation of the hemoglobin. Then we said that the deoxy state is also known as T state, and oxy state is also known as the R state. These two states are interconvertible depending upon the condition. And then when oxygen binds to the hemoglobin, and we said that the iron atom goes into the plane of the 4-pi ring, that in turn pulls the histidine residue that is a part of the globin chain, and as a result the conformational change takes place, and this conformational change in turn causes the uh, rotation of one pair of alpha beta dimers with respect to the other pair of alpha beta dimers, and as a result all the salt bridges would be progressively cleaved, and then only the O2 enters into the other subunits. First point, I just wanted to make it clear that the first oxygen molecule that enters into the hemoglobin is at the alpha subunit, because in beta subunit the O2 entry is blocked by the valine residue, but upon oxygenation of alpha subunit, as I already said that there is a conformational change takes place. That conformational change would be transmitted to the all the subunits, and as a result all these salt bridges would be progressively cleaved. Now all these subunits have the freedom to rotate. As a result, the other beta subunits also can bind to the oxygen. So that's what the then in order to understand the hemoglobin cooperativity, we have already explained the hemoglobin oxygen dissociation curve. This we have already said that the oxygen dissociation curve is the graphical representation of partial pressure of oxygen versus the hemoglobin saturation with the oxygen. That is the percentage of the hemoglobin saturated with the oxygen. And as I already said that when hemoglobin, when it moves to the lungs where the partial pressure of oxygen is 100 tar, all the hemoglobin molecules would be loaded with the oxygen. So that means the 98% of the binding sites are filled with the oxygen. When it moves to the tissues and its saturation drops to the lower level, as a result, the hemoglobin unloads its bound oxygen. Then, as I said that the hemoglobin, the O2 dissociation curve for the hemoglobin is a sigmoidal, whereas for myoglobin it is a hyperbolic, and this clearly indicates that the hemoglobin has the lower affinity towards oxygen compared to the myoglobin. That means myoglobin has a higher affinity towards oxygen, but it is not good in releasing like hemoglobin. And this you can also clearly see here that, as I already said, that the hemoglobin has the P50 value 26 tar. That means at a 26 partial pressure of oxygen, 50% of the binding sites would be loaded with the oxygen, or the 50% of the O2 binding sites in hemoglobin is occupied by the oxygen. Whereas in order to see the same 50% saturation, myoglobin doesn't require 26 star, it requires only 2 or 2.6 star. That means the myoglobin has the higher oxygen affinity compared to the hemoglobin. So then we, in order to understand, then we also discussed about that the in deoxygenated state and the oxygenated state, these two states are interconvertible in the sense, when the in deoxygenated state, as I already said that the Deoxyhemoglobin has the 2,3-BPG binding pocket between the two beta subunits. You can see here this is one beta subunit and this is another beta subunit. It has the 2,3-BPG binding pocket. So that means in the absence of the oxygen, 2,3-BPG sits in its pocket and stabilizes the T state, that is the tensed state or the taut state, or we call it as a deoxy state. So when upon O2 binding to the one subunit conformational change takes place in that subunit, then that conformational change would be progressively, the further conformational change would be transmitted to the other subunits. As a result, all these 8 salt bridges would be progressively cleaved. In addition to this, the 2,3-BPG pocket collapses. As a result, the oxygen binding takes place. As a result, the T state becomes R state. So this is about the what exactly happens when oxygen binds to the hemoglobin. Now, as I already said that what is the specialty of the hemoglobin, why it is a good cargo or the vehicle to transport oxygen from lungs to tissues. So to understand that, we have compared the oxygen dissociation curve for the hemoglobin and also the myoglobin, as well as the hypothetical non-cooperative protein. So this is not a real protein, it is only hypothetical protein. And as I already said that hemoglobin, when it moves from the lungs to the tissues, that is where the partial pressure of oxygen is 20 tar, it can unload 66% of its bound oxygen, whereas hypothetical non-cooperative protein can only release 38% of its bound oxygen. It indicates that the hemoglobin is a good vehicle to transport oxygen, and because of its cooperativity, and whereas the non-cooperative protein only contribute 38% of its bound oxygen. And of course, the myoglobin can only contribute to 7%. It can only release 7% of the bound oxygen at the 20 partial pressure of the oxygen tension. So obviously, the myoglobin is not a good cargo to deliver the oxygen to carry from lungs to the tissues. That's why nature has chosen hemoglobin as the good cargo to transport oxygen from the lungs to the tissues. Then we discussed about the what exactly happens with this. We have already said that when upon oxygen binding, the iron atom moves into the plane, and that which in turn pulls the proximal histidine residue, and this histidine, as a result, this histidine is the part of the helix, the either alpha subunit or the beta subunit, or the beta 1 or beta 2 or alpha 1 alpha 2. As a result, and as a result, when the iron atom moves into the plane and the histidine moves, as a result, all the salt bridges are progressively cleaved, and the one set of alpha beta dimer rotates to with respect to other side 15 degrees. As a result, all the 8 salt bridges would be progressively cleaved. As a result, the hemoglobin, all the subunits have the freedom for the rotation. So that means it switches from the T state to the R state. And then that's we have already explained here that with progressively you can see here that all these wavelengths are nothing but the salt bridges. As with oxygenation, this is the first oxygen molecule, and the second oxygen molecule, third, and fourth, and on so on. So that means as the O2 binds to one subunit, all the salt bridges will be progressively cleaved. As a result, the hemoglobin, the affinity towards oxygen increases further. That's what we have already said, what is meant by the cooperativity at the beginning. And then we discussed about the hemoglobin not only transport oxygen, and it also transports the carbon dioxide. And for that, we have already explained this diagram. The rapidly metabolizing tissues produce carbon dioxide, and this carbon dioxide enters into the circulation, which in turn enters into the red blood cell. And then this carbon dioxide undergoes hydration by the carbonic anhydrase enzyme and forms the carbonic acid. This carbonic acid further dissociates into the bicarbon HCO3 minus and the H plus ion, and this HCO3 minus and H plus ions, the HCO3 minus again further comes out of the red blood cell, that is into the plasma, and in order to counterbalance the charge across the membrane, red blood cell membrane, one Cl minus ion enters into the RBC. That means the HCl minus, HCO3 minus, and Cl minus enters across the membrane in opposite direction by a antiport mechanism. So this movement of HCO3 minus and Cl minus in opposite direction across the red blood cell membrane is also known as the chloride shift or the Hamburger's phenomenon. Then now this HCO3 minus has the higher solubility in the plasma. Therefore, this HCO3 minus would be transported in the plasma, in the watery blood, and when it goes to the near the lungs, again this HCO3 minus again re-enters into the red blood cell, and where again it undergoes where it is again converted into the carbon dioxide and the water by the same enzyme carbonic anhydrase, and this carbon dioxide would be further exhaled during the from the lungs. So this is the process that takes place, and so since the carbon dioxide that is produced in the rapidly metabolizing tissues, mostly 85% of the carbon dioxide is transported in the watery blood in the form of bicarbonate that is outside the red blood cell, and only 15% of the carbon dioxide is only transported in the carbamate form, in which the hemoglobin, in which the carbon dioxide, you can see here the carbon dioxide is forming the carbamate with the amino terminal of the alpha or the beta chains. And so that means the carbon dioxide, 15% of the carbon dioxide is only transported in the form of carbamate, but 85% of the carbon dioxide is transported in the bicarbonate form, and only where lesser amount is transported in the gaseous form itself. So this is about the carbon dioxide. As I already said that when the near the tissues, the carbon dioxide concentration is more. So therefore, this carbon dioxide, when it forms a carbamate with a carbamate by binding to the amino terminal of the alpha or the beta chain, and this creates a negative charge, and this negative charge, which in turn favors the formation of the salt bridges. You can also see here that the amino, these CO minus is involved in the salt bridge formation. So that means again it stabilizes the T state. So when there is a higher carbon dioxide tension, the carbamate is formed and the T state is stabilized. Therefore, the T state means the hemoglobin has to unload its bound oxygen. So that's why near the tissues, because of the higher concentration of the carbon dioxide, that in turn that in turn pushes the hemoglobin to go into the T state. As a result, the hemoglobin has to unload its bound oxygen. And then we discussed about the Bohr effect, and this is related to the hemoglobin affinity towards oxygen with reference to the pH. As we have already said that the pH near the lungs is 7.4 and the pH near the tissues is 7.2. So that means when hemoglobin moves from lungs to the tissues, there is a change in the pH from 7.4 to 7.2. So this reduced pH means there is an increased H plus ion concentration, and this increased H plus ion concentration in turn causes the salt bridge formation, facilitates the salt bridge formation by protonating the histidine 146 in the beta chain. As a result, the histidine 146 forms the salt bridge with the aspartic acid 94 on the other beta subunit. With the other beta subunit, as the result, this T state would be stabilized. So that means when the carbon dioxide concentration is more, the T state is stabilized. When the H plus ion concentration is high, again these H plus ions in turn protonates the the histidine 146, which in turn favors the salt bridge formation between the aspartic acid and the histidine 146. As a result, again, the T state is stabilized. Therefore, the hemoglobin has to unload its bound oxygen. So this is the molecular basis of the Bohr effect, and then we discussed about the effect of 2,3-bisphosphoglycerate. So 2,3-bisphosphoglycerate, as I already said, that it is an anionic molecule, and this 2,3-bisphosphoglycerate is formed from the 1,3-bisphosphoglycerate in the glycolytic pathway, and and you can see here under normal condition, as I already said, that the glucose undergoes glycolysis during in a 10 sequential steps, and the pyruvate would be formed when there is a less amount of oxygen, when the oxygen tension is less. In addition to forming the pyruvate, some amount of the 1,3-bisphosphoglycerate would lead to the formation of 2,3-bisphosphoglycerate by Loberang-Rappaport pathway. And now, this 2,3-bisphosphoglycerate is anionic molecule, as I already said, that it binds to the hemoglobin in a deoxygenated state. As a result, the two beta subunits would be cross-linked, and which in turn stabilizes the T state of the hemoglobin. As a result, again, the hemoglobin affinity towards oxygen reduces, and the hemoglobin has to unload its bound oxygen. So please carefully observe here. In the lungs, the partial pressure of oxygen is 100 tar. In the tissues, the partial pressure of oxygen is 20 tar. When red blood cell moves from the lungs to the tissues. So because of the lower partial pressure of oxygen, the glucose, the red blood cell synthesizes more and more the 2,3-bisphosphoglycerate because the partial pressure of oxygen is very less here, and therefore this 2,3-BPG in turn binds to the 2,3-BPG binding pocket. As a result, the T state would be stabilized, which in turn favors the release of oxygen near the tissues. And as I already said, that this 2,3-bisphosphoglycerate binds to the between the two beta subunits. This you can clearly see here, that the histidine 143 and lysine 82 and histidine 2, and also the amino terminal region of the amino terminal of the beta subunit. All these are involved in cross-linking the interacting with the 2,3-bisphosphoglycerate, because all these are positively charged amino acids. So this is the one beta subunit, and this is the another beta subunit, and this is the 2,3-PPG molecule. So this 2,3-PPG molecule interacts with the positively charged residues present between the two beta subunits and makes a cross link. As a result, the T state would be stabilized. So therefore, when the 2,3-PPG concentration is more and the T state is stabilized, as it is, when the T state is stabilized, obviously the hemoglobin has to unload its bound oxygen. So this is about the effect of 2,3-bisphosphoglycerate. And in morning, I have forgot to mention one point, that the fetal hemoglobin has the higher O2 affinity than the maternal hemoglobin, and this we will explain later when I explain about the different types of hemoglobins. Then I think with this, I think up to here we have explained. Then now coming to then of course we have also said that the other allosteric modulators, such as the temperature also plays an important role in the hemoglobin O2 affinity. As I already said, that increased temperature also in turn increases the basal metabolic rate, as a result, which in turn increases the 2,3-BPG synthesis. As a result, hemoglobin affinity towards oxygen reduces. Therefore, the O2 dissociation curve moves towards right. So as I already said, that decreased temperature, increased carbon dioxide concentration, increased BPG, increased H plus ion concentration, all these would allow the dissociation curve to move towards the right. Whereas decreased temperature, decreased partial pressure of carbon dioxide, decreased this was for 2,3-BPG, and decreased H plus ion concentration shifts the O2 dissociation curve towards right. So when the O2 dissociation curve shifts towards left, and it means that the hemoglobin affinity towards oxygen increases. When the O2 dissociation curve moves towards right, means the hemoglobin affinity towards oxygen decreases. So then, and this is one, and I think I at the beginning I should have explained this one. The basic difference between the alpha subunit and the beta subunit. You can see here that the hemoglobin, as I already said, that it has two alpha and two beta subunit, and the we have already explained the alpha subunit has 141 amino acids, beta subunit has 146 amino acid. The C-terminal amino acid in the alpha subunit is the arginine, whereas the beta subunit it is histidine, and the N-terminal amino acid is valine in both the cases. And the alpha subunit has 8 alpha helices, beta subunit has the 8 alpha helices. And in case of alpha subunit, the heme pocket that is adequate for the O2 entry, as I already said, that if this is the heme pocket, and this is the O2 binding site, and this heme pocket is sufficient for the O2 entry, whereas the O2 entry in case of beta subunit, the O2 entry is blocked by a valine residue. When binding of oxygen to the alpha subunit, as I already said, that the conformational change takes place. That conformational change would be transmitted to the remaining alpha and beta subunits. As a result, all the 8 salt bridges would be broken and cleaved, and which in turn moves the valine from its position. As a result, O2 can easily enter into the beta enter and bind to the beta subunit as well. And so this is about this one. So now to make it this concept a little more clear, I will just for example I just wanted to also draw a few O2 dissociation curves. We have already said that effect of pH on O2 dissociation curve. So as I already said that as the pH decreases, this is the partial pressure of oxygen, this is the percentage of hemoglobin O2 saturation. So this is the sigmoidal curve, and as I already said that as the pH decreases, let us say this is the 7.4, pH 7.4, as the pH decreases, the O2 dissociation curve moves towards right. So it is, for example, pH 7.2. As the pH increases, the O2 dissociation curve moves towards left. This is pH 7.6. So when the O2 dissociation curve moving right means it has a lower affinity towards oxygen. When the O2 dissociation curve moves towards left means the hemoglobin has a higher affinity towards oxygen. This you can clearly see that. Once again I am just saying that if this is 100% saturation, and here you can see clearly here that at pH 7.6 the P50 value is lower compared to the pH 7.4 and the pH 7.2. That means lower P50 means, and it has the higher O2 affinity. That means it can, at pH 7.6, the hemoglobin molecule can achieve 50% saturation at the lower partial pressure of oxygen, but the same 50% saturation can be achieved at a higher O2 tension at the lower pH. That means at that lower pH, the hemoglobin has the lower affinity towards oxygen. So that's why, so with reference to the pH, with reducing the pH, the O2 dissociation curve moves towards right. And this is about this one. Then coming to the carbon dioxide. Coming to the effect of carbon dioxide, these diagrams, it will only give you the clarity, partial pressure of oxygen in tar, and the percentage of hemoglobin O2 saturation. So again, again here, for example, with increasing the carbon dioxide concentration, the O2 dissociation curve also with increasing the carbon dioxide concentration, the O2 dissociation curve also moves towards shift towards the right. So this is about with reference to the carbon dioxide. You can just for example, for the sake of understanding, you can say that 20 tar or the 40 tar or the 50 tar. So with increasing the carbon dioxide concentration, the hemoglobin O2 dissociation curve moves towards the right. So that's why near the tissues, the carbon dioxide concentration is more, and increased carbon dioxide concentration in turn stabilizes the T state. As a result, hemoglobin affinity towards oxygen reduces, hence the hemoglobin has to unload its bound oxygen. So this is about this one. Now coming to the temperature, as I already said that with increasing the temperature, the partial pressure of oxygen versus percentage of saturation, with increasing the temperature, the basal metabolic rate also increases, which in turn increases the synthesis of the 2,3 BPG, 2,3 BPG. So therefore, so we think, for example, if it is 10 degrees centigrade and if it is 20, this is the 38, and if it is 43 degrees centigrade. So with increasing temperature, the O2 dissociation curve shifts towards the right. That means it has the lower affinity towards the oxygen. Hemoglobin has the lower affinity towards the oxygen. Then last one is the BPG. BPG concentration, that is the partial pressure of oxygen versus percentage of saturation. And again, the with increasing the concentration of 2,3 BPG, the O2 dissociation curve also moves towards the right. So for example, if the BPG concentration is 1 millimolar, BPG concentration is 1 millimolar or 5 millimolar or 8 millimolar. So that means with increasing the BPG concentration, again O2 dissociation curve moves towards the right. So that means hemoglobin affinity towards oxygen decreases. So these are these things you need to remember, how the O2 dissociation curve moves towards right or left, and in a given scenario. For example, in certain cases, for example, you can just see here that if I give you the O2 dissociation curve, and if I ask you to match this O2 dissociation curve with the conditions given here, partial pressure of oxygen in tar and the percentage of hemoglobin saturation, and let us say that this A condition A is pH 7.4 and no carbon dioxide. B, these are the conditions. B, pH 7.2, no carbon dioxide. C, pH 7.2, 40 tar of carbon dioxide. So now it is very clear when you take A and B, so A has pH 7.4, B 7.2. That means as the pH reduces, hemoglobin dissociation curve moves towards right. So that means definitely, obviously, this would be B and this would be A. Now compared to B and C, B has pH 7.2, no carbon dioxide, but C has pH 7.2, but the 40% of 40 tar of the carbon dioxide. Obviously, so in addition to the pH 7.2, if it has a 40 tar of carbon dioxide, again the O2 dissociation curve still moves towards right. So that means this would be C. So in the same, that means we can also give, we can also add other parameters like BPG or and so on. So I am just giving you the clue that in exam you can expect any this type of water dissociation curves, and you should understand that the given conditions and which the dissociation curve is appropriate to match the conditions given in the uh, uh, conditions given. So this is about this one. Now coming to the we have the that is a last topic, that is hemoglobinopathies as well as the normal and abnormal hemoglobins, and this is you can see here that so there are different types of hemoglobins observed, and we are not going to discuss all. There are many. We will discuss only very few. And here, see the hemoglobin A. First one is the hemoglobin A. Hemoglobin A is nothing but it is the normal adult hemoglobin, and as you can see here what we have discussed so far, that is the two alpha chains and the two beta chains. This is a normal hemoglobin, and this HbA1 constitutes 90 to 95% in normal adults. So that means 90 to 95% of the hemoglobin present in normal adults is nothing but the hemoglobin A1. And now coming to the hemoglobin F. So as I already said that hemoglobin F is the fetal hemoglobin, and this fetal hemoglobin has two alpha chains and two gamma chains, two alpha chains and the two gamma chains. And this point you need to remember that the alpha chains, two alpha chains and two gamma chains, that means the beta chains are replaced by alpha gamma chains in case of fetal hemoglobin, but the beta chains and the gamma chains are 72% identical, but except a few differences in the amino acid composition. And um, so one important difference between the gamma chain and the beta chain is that in the beta chain, in 143 amino acid is histidine, that is, it is involved in the salt bridge formation, so it is involved in the binding with the 2,3-BPG, whereas in case of gamma chain, the 143 amino acid is serine. So that means the positively charged amino acid present in a normal adult hemoglobin is replaced with a polar uncharged amino acid, that is a serine. So that means, as I already said, that in in adult hemoglobin it has the two alpha chains and two beta chains, and the beta chains have the basic amino acids, mainly the histidine, histidine 143, histidine 2, and lysine, and all these are involved in making the 2,3-BPG pocket. So when the histidine, in the case of gamma chain, the 143 position, the histidine is replaced, instead of histidine, the serine is present. So therefore, since the serine is present, 2,3-BPG cannot bind to the fetal hemoglobin as efficiently as it can bind to the adult hemoglobin, because the gamma chains are identical with the beta chains, but they have the certain differences. And the main difference is that in 143 position, in case of gamma chain, serine is present, whereas in case of adult hemoglobin, the histidine is present. This histidine is involved in the formation of the 2,3-BPG pocket. As a result, that facilitates the 2,3-BPG binding, whereas in case of the fetal hemoglobin, 143 amino acid is serine. So therefore, 2,3-BPG cannot bind to the fetal hemoglobin like it binds to the adult hemoglobin. It can bind, but it cannot efficiently bind like it binds to the adult hemoglobin. Therefore, when the 2,3-ppg is not efficiently binding to the fetal hemoglobin, means the fetal hemoglobin has higher O2 affinity than the adult hemoglobin. So that's why the fetal hemoglobin is uh, able to extract oxygen from the maternal blood because of the higher O2 affinity. So if you look at the O2 dissociation curve for the fetal hemoglobin and the adult hemoglobin, if this is the O2 dissociation curve for the hemoglobin A1 adult hemoglobin, the O2 dissociation curve for the fetal hemoglobin would be like this. This is the hemoglobin F. That means this is the fetal hemoglobin. That means it has the higher O2 affinity compared to the maternal hemoglobin or the maternal hemoglobin. This we can say that maternal hemoglobin, that is nothing but the hemoglobin A1. So this is about the hemoglobin F. Now coming to the hemoglobin A2, this hemoglobin A2, it is the minor component of the adult hemoglobin, and it is present only 2.5% out of 100%, and this is the hemoglobin A2. Coming to the hemoglobin A3, hemoglobin A3, it is also present in 3 to 10% in the total hemoglobin, and it is a modified hemoglobin mainly present in the older red blood cells. Older red blood cells. So this is the hemoglobin A3. Then hemoglobin A1C. This is a very important. This is also known as the glycosylated hemoglobin, and this glycosylated hemoglobin, in the glycosylated hemoglobin, and in this, the the amino terminal side of the beta subunit you can also see here. So the beta chain has the, as we have already said, that the valine residue is present in case of alpha as well as the beta subunit at the amino terminal, and the sugar covalently linked to the valine residue at the amino terminal side of the beta chain. As a result, the glycosylated hemoglobin is formed, and this glycosylated hemoglobin is only 3 to 5% in the normal individuals, whereas in case of diabetic patients, its level, its concentration goes up from 6 to 15% of the total hemoglobin. That means this glycosylated hemoglobin, in the glycosylated hemoglobin, the glucose or the sugar molecules covalently linked to the hemoglobin at the amino terminal region of the beta chain. As a result, the glycosylated hemoglobin is formed, and the coupling of the sugar to the amino terminal region of the beta chain is a non-enzymatic process. So therefore, it takes a longer time. So that's why when this glycosylated hemoglobin doesn't change with overnight. In the sense, when before going to the clinic, if we consume any diet which has a high concentration of sugar, the postprandial or the fasting blood glucose tend to change, whereas the glycosylated hemoglobin doesn't change. In the sense, because the coupling of the glucose to the hemoglobin takes a longer time, because it is a non-enzymatic process and it takes a longer time. So that's why this glycosylated hemoglobin is used as a marker to diagnose the diabetes, because it tells us that the glycosylated hemoglobin will give us the overall concentration of glucose over a period of 2 weeks in the circulation. As I already said, that the postprandial or the post fasting blood glucose or the postprandial blood glucose may change depending upon the type of the diet we consume before going to the clinic, or depending upon the type of exercise we do. But the glycosylated hemoglobin doesn't change, because the formation of the glycosylated hemoglobin is a slow process. It is a non-enzymatic process. For the coupling of the glucose to the hemoglobin, it takes the weeks. So that's why this glycosylated hemoglobin is used as a marker for diagnosing the diabetes. So in case of normal individuals, it is only 3 to 5%. In case of diabetic patients, it goes from 6 to 15%. And these are the about different hemoglobins. In addition to this, we have one abnormal hemoglobin, that is a sickle cell hemoglobin. In sickle cell hemoglobin, in sickle cell hemoglobin, yeah, in sickle cell hemoglobin, as you can see here that in sickle cell hemoglobin and the glutamic acid that is present in the sixth position of the beta subunit is replaced with a valine, and because it is a mutation, so when the glutamic acid is replaced with a valine, and here we need to understand that the glutamic acid is a polar charged amino acid, whereas the valine is a non-polar amino acid. When a polar charged amino acid is replaced with a uncharged the hydrophobic amino acid, that brings a conformational change in the hemoglobin structure, and as a result, you can see here that as a result that creates a sticky patch on the sickle cell hemoglobin. You can see here that creates a sticky patch on the sickle cell hemoglobin, and in deoxygenated state, the sickle cell normal hemoglobin, this you can see here, this is the normal hemoglobin in the oxygenated state, and this is the deoxygenated state, and this is sickle cell hemoglobin. So because of the replacement of glutamic acid by valine, and that brings a conformational change, and that creates a sticky patch on the surface of the beta subunit. And so in deoxygenated state, these sickle hemoglobin molecules, you can see here in deoxygenated state, the sickle cell hemoglobin molecules forms a long fibrous precipitate, because in deoxygenated state, the sickle cell hemoglobin has the sticky patch as well as the pit. So therefore, when the many hemoglobin molecules come closer, the sticky patch on one hemoglobin molecule exactly fits into the pit of the another hemoglobin molecule, because the alpha subunit has the pit, whereas the beta subunit has the sticky patch. So therefore, in deoxygenated state, the sickle cell hemoglobin polymerizes and forms a long fibrous precipitate, as you can see in the this is the electron microscopic picture of the sickle cell hemoglobin. When it is polymerizes, it forms a long fibrous precipitate, and this formation of this long fibrous precipitate, which in turn destabilizes the structure of the RBC, because this RB, when it is a hemoglobin, deoxyhemoglobin in sickle cell hemoglobin polymerizes, which in turn uh, uh, forms a long fibrous precipitate that in turn stabilizes the shape of the RBC. As a result, hemoglobin goes into the sickle shape. The RBC goes into the sickle shape, and this sickle shaped red blood cells cannot freely move in the circulation. They causes the occlusion or the obstruction of the blood vessel. As a result, it leads to, and these in addition to this, these sickle cell hemoglobin sickle cell red blood cells are highly vulnerable to the lysis, and it also impairs the passage through the blood vessels. And this is about the sickle cell hemoglobin, and here it is because of the mutation, and in normal hemoglobin, in beta subunit, the history of glutamic acid is present in the sixth position, and whereas in sickle cell hemoglobin, instead of glutamic acid is a valine is present, and so therefore when the charged amino acid is replaced with a non-polar hydrophobic amino acid, that creates a sticky patch, and which in turn in deoxygenated state that polymerizes and forms a long fibrous precipitate, and which in turn destabilizes the shape of the RBC, and becomes the sickle shape. And these sickle cell shaped red blood cells are highly vulnerable for the lysis, and they cannot pass through the blood vessels easily, and they causes the occlusion or the blockage of the blood vessels, which in turn increases the viscosity as well as and causes the inflammation as well. So this is about the sickle cell hemoglobin. So this is about the abnormal hemoglobin.